Whereas Chapter 9 dealt mostly with inheritance patterns and looking at how chunks of information are passed on, in Chapter 10 we're going to look at genetics from a little bit different perspective. We're going to look at it from that chemical or molecular level. Structurally, how is DNA built? How does DNA function in terms of building different proteins? And we certainly know that DNA is that genetic material. It is that chemical that provides information on when to build proteins, information on how to build those proteins, which amino acids go in the correct order, so that we get a functioning protein for over 250,000 different proteins that we need as humans. Now we know that as we look at our DNA structure, our monomers, our basic building blocks of DNA, are nucleotides. And these nucleotides each have a sugar component, and the sugar is deoxyribose. That's where DNA, deoxyribonucleic acid, gets its name, is from that sugar. Each nucleotide has a nitrogenous base, which is basically a ring structure with lots of carbons and nitrogens, and each monomer has a phosphate group as well. Now there are only 4 different nucleotides, there are only 4 different building blocks that we connect into long chains to make up DNA. Each of these nucleotides has the same sugar and phosphate, but what's different is that nitrogenous base structure. And our 4 nucleotides are adenine, thymine, cytosine, and guanine, or as we'll abbreviate them throughout the chapter if you've looked at your book already, A, T, C, and G. When our cells build DNA, then, we're going to take these nucleotides and we're going to connect them together into long chains. And so if we look at the figure there on the right, that's a single nucleotide, the phosphate group, the sugar group, and then nitrogenous base. This particular ring structure happens to be T, or thymine. Then our cells are going to connect these together into our long chains. The phosphate of one nucleotide is connected to the sugar of the nucleotide above it. And so if we look at the center there where that thymine is, that phosphate is connected to the sugar of that cytosine, or C, above it. In turn, the phosphate of the cytosine is connected to the sugar of the adenine above it. And so what we get are these alternating phosphate and sugar chemical groups, which are sometimes referred to as the sugar-phosphate backbone of DNA, because it's actually holding the DNA together. And then the nitrogenous bases, the part that's different for each nucleotide, are sticking out to the side there. And here we can see examples of A, C, T's, and G's. In last chapter we looked at those alleles, those alternate forms of genes that contain slightly different versions of information. Well, it turns out the different information that's contained in DNA is contained in the sequence of the nucleotides. It's the order of those adenines, thymines, guanines, and cytosines that convey genetic information or protein building information to the cell. And so a particular gene contains information, a particular sequence of nucleotides that's going to provide information on how to build a protein in our cell. And then, again, different alleles or versions of that gene are going to be slightly different sequences of A, T, G's, and C's. We know that DNA isn't made up of just one nucleotide chain, but it's made up of 2, and those 2 chains tend to form a spiral staircase kind of shape, or a helix. And that's where our double helix structure comes in, which was first discovered back in the 1950s by Watson and Crick. And so the double part of the double helix refers to those 2 separate chains of adenines, thymines, guanines, and cytosines. Remember, our sugar-phosphate bone holds each chain together, and each chain has those bases sticking out. Well, those bases then will bind with each other, chemically actually stick together, and act as sort of a zipper and hold those 2 chains of nucleotides together. And once they're bound together, they will kind of form that spiral or twisting shape, that helical shape. Now, it turns out that these bases are not going to just randomly pair or bind with each other, but because of their chemical structure, we're going to see what's called complementary base pairing. And this complementary base pairing is absolutely critical for how DNA gets copied and how it provides information to cells. Adenines will only chemically bind with thymines, and vice versa. Thymines will only bind with adenines because, again, of their chemical structure. Likewise, our other pair, guanines will bind with cytosines, cytosines with guanines. We won't see adenines binding to guanines or adenines to other adenines because of their chemical structure. A's with T's, G's are always with C's. So if we take a look at that chemical structure, in the center there, here's our 2 strands of nucleotides, our phosphate sugar backbone holding both strands together, and our A's are pairing with T's, G's are pairing with C's, acting to zip that together. And you may notice that the chain on the right is upside down compared on the left. That's called antiparallel. Our 2 strands of nucleotides are kind of heading in opposite directions there, but that's the normal part of our structure of DNA. Once those complementary bases will bind together, then we'll have that twisting shape, and we can see our double-stranded helix example on the left in what's called a ribbon model, kind of showing our sequence of A, T, G's, and C's in our DNA. On the right is more of a 3-dimensional computer model of what the DNA might actually look like. And so, for example, if one of my nucleotide strands had this sequence in it: adenine, adenine, thymine, thymine, cytosine, guanine, 3 guanines in a row, ACT, and then TCC. If that were the sequence of nucleotides in one strand, I can certainly predict then what is the sequence going to be in the other strand because of my complementary base pairing. So pause for a moment and just write in what is the sequence of that other strand. So A's and T's are going to pair up, G's and C's are going to pair up. So our other strand would be TTA, AGC, CCC, TGA, and AGG. So because of that complementary base pairing, there's only one sequence in one strand that would pair up with the sequence in the other strand. If we look at the structure of DNA then, it's really pretty simple. We've only got 4 nucleotide building blocks, A, T, G's, and C's. Our cells connect them into 2 chains of nucleotides. Those nucleotide pairs bind together then. A's always pairing with T's and G's with C's. And so not a lot of complexity here. Compared to something like proteins, for example, proteins have 20 different amino acid building blocks. Sometimes proteins may be made up of 1 polypeptide chain of amino acids, sometimes 2, sometimes 4. And so a lot more complex structure when we look at proteins. And in fact, for a long time in the 30s and early 40s, when biochemists were trying to figure out what is that heredity or genetic material, a lot of biochemists ruled out DNA because DNA was too simple. And they reasoned that a simple chemical cannot contain enough information on how to build something more complicated. Okay? Today we know, obviously, despite the fact that DNA is simple, it is that genetic material. And the reason that DNA can contain protein building information that's more complicated is just by the sheer volume or amount of DNA that we have. And as humans, we've got 46 chromosomes or pieces of DNA in our cells, and the average chromosome size is 140 million nucleotide pairs long. Okay? Now there's a lot of variation. Chromosome 1 is really large, and chromosome 22 and the Y chromosome are really small. But if we average them out, about 140 million nucleotide pairs of information for every single chromosome. Our heredity material must have 2 very important functions. First, it must be able to replicate or copy itself, because every time our cell undergoes cell division, we have to copy our DNA ahead of time and pass that genetic information on. And so one important function of DNA is this replication or self-copying aspect. The other function of our hereditary material obviously is to provide that protein building information. Okay? So we're going to look at replication first, and then we'll look at the protein building aspects. Now, because we have so much DNA, we just mentioned how much DNA we have, and because that genetic information is so important, if we have any mutations or mistakes, that may impact our health in a very big way if we don't build the correct protein and we have a protein that doesn't function correctly. And so because of these issues, this DNA replication has to be accurate. We have to make mistakes at a very, very low rate. And in fact, that's the case. On average, mistakes are made about 1 every 100 million or more gametes that we produce. We get a mistake in one of our genes. And so that's a pretty good accuracy when we look at DNA replication. And the reason this replication can be so accurate is because of the chemical metabolism or process of our copying of our DNA. And that's done by what's called semi-conservative replication. And in semi-conservative replication, our name is kind of giving us a hint. Some, or semi, of the original DNA is going to be conserved or reused when we replicate or copy our DNA. And in particular, what happens is that original DNA is going to serve as a template to build the new copies of DNA. And so by using our original DNA as that copying template, again, it's going to increase our accuracy of our process. And if we take a look here at just a small chunk of our DNA, here's our original DNA with a sequence of A, C, G, A, T. If I look at the left-hand strand, and of course the right-hand strand, A's and T's pair up, G's and C's pair up. So in our semi-conservative replication, enzymes will unzip and peel apart our 2 strands of DNA. Once that happens, other enzymes will come in and pair up the matching bases. Again, A's with T's and G's with C's. So that when we're finished, now on the right there, we've got 2 identical copies of DNA. Notice they both have the sequence of A, C, G, A, T, the same as the parent. But each of those 2 copies has 1 of the original strands in blue and then 1 new strand. And so that's the idea of semi-conservative replication. We're conserving or reusing that original DNA as a template to make sure we build the correct sequence with our new strand of DNA. And DNA replication is an extremely complex chemical pathway. There are many, many enzymes involved. In addition to just simply opening and unzipping the DNA, enzymes have to start the copying process. We have proofreader enzymes that will check for mistakes, any mismatches. For example, if an enzyme sticks a T across from a C, that's not a match. And so proofreader enzymes can remove that T and put a G in across from the C, the correct nucleotide base. And so a very, very complex pathway. We're certainly just thinking about this as an overview of how our DNA is going to get copied. And we understand this process extremely well. In fact, we can use our knowledge of DNA replication and actually copy DNA in a test tube in an artificial setting using a process called PCR, which stands for polymerase chain reaction. So here's an animation showing our DNA replication. Our enzyme is moving down that double-stranded helix, unwinding, unzipping it. Proteins will bind to each strand as it goes to keep the strands from simply rezipping or binding behind it. Other enzymes are copying the DNA. And because one strand, remember, was upside down compared to the other, our copying is going to be a little bit different on the two strands. On one strand, we copy just one long piece. On the other strand, we tend to copy in small little chunks, which are called Okazaki fragments. So our first function of DNA is that replication, or being able to copy itself. Our second function of DNA, obviously, is to make proteins. And to make proteins, our DNA, remember, our flow of information gets converted into RNA, and then that RNA is actually going to do the building of the proteins. And anytime we have DNA converted into protein, we have this flow of information activated or turned on. That's referred to as gene expression. Our genes are expressed when they're producing proteins. And so before we jump into our function here of DNA building, having this protein building function, we need to look at RNA, sort of the go-between or the in-between molecule here. And RNA chemically, which stands for ribonucleic acid, is very, very similar to DNA. Not surprisingly, they're very similar chemicals here. And I just want to point out a couple of exceptions. One exception is DNA is made up of A, T, Gs, and Cs. But in RNA, uracil replaces thymine. And so instead of having a thymine, RNA is made up of A, U, G, and Cs. Now, uracil chemically is similar to thymine, and it will bind to adenine. And so A's and U's pair up, G's and C's pair up if we're dealing with RNA. Our sugar in our nucleotide is different. Our sugar is ribose instead of deoxyribose. And most RNAs are single-stranded, and so RNAs tend to be much smaller. So we mentioned our average piece of DNA is double-stranded, 2 strands of nucleotide that is 140 million nucleotide pairs long. RNA, on the other hand, is just one strand of nucleotide, and the average pieces of RNA probably average more in the tens or hundreds of thousands as opposed to millions of nucleotide bases long. So if we just compare our DNA and RNA kind of side by side, again, RNA is going to be single-stranded. The sugar is ribose instead of deoxyribose, and those sugars are extremely similar chemically. Not surprisingly, deoxyribose is missing an oxygen, as is highlighted in the picture there. And then the nucleotide bases are going to be slightly different. DNA made up of A, T, Gs, and Cs. RNA made up of A, U, Gs, and Cs. To build a protein going from our DNA to RNA to protein, then, there are 3 different RNAs that are going to play a role in this protein building process, and the RNAs are just named based on their job or the role that they're going to play in our protein building. So first up here is mRNA, which stands for messenger RNA. And if we think about the protein building process, the genetic material to store up the information is in the nucleus. Proteins, though, are going to be physically built out in the cytoplasm. And so at some point we're going to have to get that genetic message from the nucleus out to the cytoplasm. And that's the role that messenger RNA plays. It's the messenger, or it's going to relay that genetic message from DNA inside the nucleus to the protein building machinery, if you will, out in the cytoplasm. And messenger RNA is made up of a long single strand of A, U, Gs, and Cs that's copied directly from the gene in the DNA. And so we can think of messenger RNA sort of hair-shaped. It's very long and thin, like a strand of hair. So if we had a gene, for example, here, when it's time to make protein from that gene will be opened up by enzymes. Okay. The top strand here is going to be ignored. The bottom strand is the template strand, and that strand is going to be used to make our messenger RNA. And so our RNA bases are going to come in to match that DNA. So our messenger RNA is not an exact copy of DNA, but instead it's a matching copy. G's pairing up with C's, U's with A's. And so here we're going to get a very specific sequence in our messenger RNA, in this case GUC, AUUC, GG, that's matching that DNA. Another important RNA is T, or transfer RNA. And the second obstacle we're going to have to overcome here in this chemical process of building proteins is that our information is stored up in the nucleotide, or the genetic language of DNA, A, T, G's, and C's, but our proteins are built with amino acids, kind of our protein language. And so at some point we're going to have to transfer our chemical language from nucleotides to amino acids, and that's the role transfer RNA plays, is it's going to help us kind of translate or change languages from one to another here. And not surprisingly, transfer RNA is made up of both amino acids and nucleotides, so that's how it's going to kind of be that go-between molecule. Now it's also a long single strand of A, U, G's, and C's, but because of the sequence of the transfer RNA, the single strand tends to fold up and bind on itself. And so instead of being long and hair-like messenger RNA, transfer RNAs are often folded up and have more of a three-dimensional sort of cloverleaf shape. And we can kind of see that here. Here's our sequence of A, U, G's, and C's that makes up the transfer RNA. And again, based on the base pairing, we've got our RNA binding with itself to make that very specific shape. And then at the one end of the molecule, which is at the bottom of our picture here, an amino acid would be attached to it. And so transfer RNA is made up of both of those different building blocks. Last up then is rRNA, which stands for ribosomal RNA. And ribosomal RNA, along with proteins, is going to form one of our cell organelles, the ribosome. And remember, the ribosome is the place for protein synthesis. This is going to give the cell a physical place to get all the right chemicals together in the cytoplasm and actually connect amino acids together and start building a protein. And so our ribosomal RNA is more of a structural component as opposed to transfer and messenger, which are more involved in the metabolic process. And so here we can see a ribosome of a typical eukaryotic cell like an animal or a human cell might have. It's made up of two distinct pieces. There's the bottom plate that's made up of a combination of ribosomal RNA and proteins, and then there's a large cap that kind of both chemically locks to that plate, which is also made up of different ribosomal RNA and different proteins. If we take a look at gene expression then, and again, gene expression just means the gene is turned on. We're going to undergo our DNA to RNA to protein process for a particular gene. In our eukaryotic cells, there are three major chemical steps here: transcription, processing, and translation. The transcription and processing both occur in the nucleus. Translation then is going to be the protein building out in the cytoplasm. So we're just going to walk through these steps again and look how our genetic information is going to provide that protein building information. First up then is transcription, and because this is our first step dealing with the DNA again, it is going to occur in the nucleus. And these are, again, very complex metabolic pathways, lots of enzymes involved, but basically these enzymes are going to take our gene, our genetic information in our DNA, and unwind and unzip the DNA, peel apart the two strands of DNA and kind of open them up. And then other enzymes are going to bring in RNA bases, A, U, G's, and C's, that pair up to that template strand of the DNA. And remember, it's a template strand that is going to provide the protein building information. So that by the time we're done with transcription, what we're going to end up with is a messenger RNA copy of the gene. So a piece of single-stranded messenger RNA that is a matching basis of our genetic information from our template strand of our DNA. So if we take a quick look at this little animation then in transcription, again, each gene has a promoter region and a termination region that just indicates where to start copying the DNA and where to stop. Our enzymes will bind to the DNA, unwind and unzip it, and as those enzymes slide down that double-stranded DNA, it's going to bring in matching RNA bases to pair up with the template strand. That second strand, in this case the top strand, is called the complementary strand. It's kind of a bystander. It's not going to be used. We're only going to copy that template strand. And if we take a look at a close-up, what we will see is that it's those complementary base pairs. A's pairing up with T's in the DNA, U's with A's, G's with C's, and so on, that's going to provide our very specific sequence of our messenger RNA that's going to be utilized then to eventually build the protein. Once that messenger RNA is complete, then while it's still in the nucleus, it's going to undergo this processing stage. Now this processing stage only occurs in eukaryotic cells, like animals and plant cells, for example. And this was a real surprise. When biologists were studying this process of protein building, we assumed that a gene contained a sequence of A, T, G's, and C's, and all of that genetic information, that gene, was important for building the protein. Today we know that that's not the case, but instead our genes are made up of alternating chunks of information, and these chunks can be a few hundred to a few thousand nucleotides long, and the chunks that provide protein building information are referred to as exons. So exons, by important information, again, I mean they contain information that we need to build our protein. And again, biologists originally thought, well, all of our gene is important information, but stuck amongst those exons, we have chunks of information referred to as introns. And these introns have no function in terms of our protein building. They're not going to be used to build the protein, even though genetically it looks just like the rest of the DNA. It's a sequence of A, T, G's, and C's here. Now, it turns out we know that these introns probably do have functions in terms of regulating genes and turning genes on and off. But in terms of building a specific protein, this intron information is not going to be used. So when we make our messenger RNA copy of the DNA, we copy both exons and introns. So during this processing stage, we need to get rid of the introns. We only want that exon information. And so in this stage, enzymes are going to go through and splice out the introns, physically cut them out of the messenger RNA, and then reassemble those exons back together. And once we have just the exons, that's referred to as mature messenger RNA. And if messenger RNA is mature, again, that means there's no introns. It only has protein building information. So if we take a look here, again, a real surprise, we've got these alternating chunks of exons and introns, protein building versus non-protein building information. So when we make our messenger RNA, in this processing stage, we're going to snip out those introns, reconnect those exons together. And so now we've got our mature messenger RNA. It's going to leave the nucleus out to the cytoplasm, and we're ready to start then actually physically building our protein and connecting amino acids together. And the final stage of our gene expression then is translation, and this is the RNA to protein stage where we are going to start building the physical protein. And this is the only step of the three that does occur out in the cytoplasm. But to get RNA information converted into protein, cells use what's called the genetic code. And the genetic code is used to convert our language here from nucleotides to amino acids. We can think of it as the cell's genetic language, and it's very similar, a lot of analogies between the genetic code and our written language. Okay? In our written language, we have letters that make up our alphabet. We've got 26 letters. We arrange those letters into words, and it's words that have meaning. For example, if I ask you to read that line right below genetic code, I don't think you would read it to me as USEDTOGETFROM, right? You would read the words, used to, get, from, okay? The same thing here with our genetic code. It turns out the cell's genetic alphabet is only 4 letters. It's A, U, G's, and C's. It's made up of these nucleotides. But when the cell builds proteins, it doesn't read every letter. Instead, the letters are arranged into little genetic words that are referred to as codons, okay? Now, one difference with our genetic code is whereas in our language, words can be any length, from 8 or 9 letters to maybe only 1 or 2 letters. In the genetic language, every codon or genetic word is the same, and they're all 3 letters long. So a codon represents 3 letters or 3 nucleotide bases in the messenger RNA. For example, the genetic word AAA, or 3 adenines in a row, would be a codon, and that would have specific protein building information to the cell in terms of which amino acid should come next. A couple things about our genetic code, since it is so integral to building our protein. First of all, the genetic code is universal. And there aren't many times all semester that I'll use the term universal, because it seems for every rule in biology there's an exception, just because there's so much diversity of life. But here's a case where we haven't found any exceptions yet. That the genetic code, which you see on the right here, is the same for all living things. So the genetic word AAA, 3 adenines in a row I mentioned. If we find where 3 A's are out there, we see 3 A's code for LYS, which stands for lysine, one of the 20 amino acids. So every time 3 A's appear in our messenger RNA, that's going to code for a lysine amino acid in a protein. And it doesn't matter whether I'm looking at a bacterial cell or one of your cells or a blade of grass cell out there in your lawn. The other thing about the genetic code is that it's a degenerate code. And by degenerate, what that means is there's lots of repetition. We've only got 20 amino acids in a protein, but if we look at our genetic code, there's 64 different words that we can come up with. And so there's more than one code or codon for each amino acid. For example, if we look at leucine, L-E-U, on our chart in the upper left there are 6 different words or codons that all have the exact same meaning. And so that's what's meant by this degenerate code. Okay? So notice, highlighted there are some punctuation words in our genetic code. All of our genes start with the genetic word AUG. and that indicates that's the start of the protein building information. Likewise, all of our genes end with a period or a stop codon, and we see there's 3 different stop codons up there, UAA, UAG, and UGA. And so all of our genes are going to start with AUG, and they're going to end with one of those stop codons, and in between are going to be lots of these other codons providing information on which amino acids should be added to build this particular protein. So our RNA to protein step, our translation, we're going to use our genetic code, and there are 3 chemical stages within translation. We've got a lot of chemicals going on here. We've got messenger RNAs with the genetic code. We've got our ribosomes where we're going to build it. We've got our transfer RNAs that are going to help build the protein. So in initiation, basically all these different chemicals are going to be brought together into one place. Once they're brought together, then we're going to undergo what's called elongation, which is actually building the chain of amino acids. We're going to add one amino acid to the chain, and then the next amino acid, and so on. And again, proteins can be made up of just 50 or 100 amino acids, or they may be made up of several thousand amino acids all hooked together. Then eventually we're going to have termination, which is the end of the protein building. So when we come to one of those stop codons, that's going to indicate no more amino acids are needed, and we're going to terminate our translation process. And transfer, or tRNA, is going to play a big role in this whole process of translation. And remember, our transfer RNA is made up of both chemicals, nucleotides, and amino acids. At one end of the transfer RNA is an amino acid. At the opposite end of the transfer RNA, there's a 3-base segment that's referred to as the anticodon. And these anticodons are AU, Gs, and Cs, part of the sequence of the transfer RNA, that match the codons. And so we've got 64 codons, or words, in our genetic code that we just saw on a couple slides ago. We've got 64 transfer RNAs that match those codons. Okay? And so here's our picture again. Again, at the bottom we have our amino acid. At the top we've got our anticodon. And these transfer RNAs are going to come in, and the anticodons will bind to the codons, and that's how we're physically going to drag the amino acids together in a place to make our protein. And so, for example, let's say my messenger RNA had this sequence: AUG, CCC, GUA, and CGC. And I'm going to put them in groups of 3 because that's what our codons or words are. Well, out there then in the cell, in the transfer RNA, is going to be transfer RNA with a codon that matches AUG. And based on our complementary base pairing, the only anticodon that would match that would be UAC. And so that codon would match. And when that transfer RNA binds there, it's going to drag its amino acid behind it. Our next anticodon would be GGG, that would pair up with CCC. Next up would be CUA and GCG. And again, these anticodons are just binding to the codons and physically moving the amino acids. So in initiation, we're going to get all these important chemicals together. And our eukaryotic ribosomes, as I mentioned before, have 2 parts to it. The bottom sort of plate, and then the large cap over the top. And the cap has 2 large spaces in it that provide information to read 2 genetic words or 2 codons of the messenger RNA at a time. Think of our ribosome sort of like a 2-car garage. It's got 2 stalls in it. Okay? So in initiation, we're going to get our different chemicals together here. And so on the left there, if we start, what's going to happen is that messenger RNA at the start codon, that AUG, is going to bind to the small portion of the ribosome, that bottom plate. Once those 2 things bind together, then that transfer RNA with the matching anticodon, which would be UAC, is going to come in and bind to that start codon. So again, we're starting to accumulate or get all of our chemicals together. Once those 3 things are chemically bound together, then the large cap of the ribosome is going to bind over the top and sort of lock everything together. And so that ribosome cap is going to lock it together. And notice that once it's locked together, our 2-car garage, our 2 spaces of our ribosome, one is filled up with the transfer RNA matching that start codon, but the second site there, the second space, is blank. And so now we can read our next genetic word, and we can start building our protein. Now that all of our chemicals together and the cap has locked everything in one place, elongation is the longest part and the major stage of our protein building, where we're going to start adding 1 amino acid at a time. And so that transfer RNA is going to come in and bind to that codon in the empty space. And so we're going to move from 1 in our figure over to 2. That transfer RNA is going to come in and bind to that codon, carrying along its amino acid. Okay? Once that amino acid then is close to the amino acid on the transfer RNA on the left, then enzymes will connect those 2 amino acids together. Okay? Then we're going to move to step 3, where that first transfer RNA that was originally there will be cut out and removed by enzymes. Then the ribosome will actually ratchet or slide down the messenger RNA moving the transfer from the right spot into the left spot. Okay? Once that happens, now we're ready to read the next codon. So the next transfer RNA will bind to it, add its amino acid to the chain, cut out the old transfer RNA, ratchet down, and just repeat that process over and over again. So if we look at the moving pieces here of initiation, that bottom plate of the ribosome is going to attach to the messenger RNA at that start codon of AUG. And so we can see those two things binding together. Then that transfer RNA with the matching anticodon, which again would be UAC, is going to come in and bind to it physically. And notice that MET, that's the methionine, that's an amino acid. So each transfer RNA has an anticodon at one end and an amino acid at the other. And now our large ribosome cap is going to bind over the top and kind of lock everything together. And physically, that ribosome surrounds the messenger RNA. It kind of actually travels through the ribosome, allowing the ribosome to slide back and forth or up and down on that particular piece of messenger RNA. So that's the end of initiation. Now we're ready to begin building the protein. So now that initiation is complete, we're ready for elongation to build our protein, add amino acids together. And so our first space there now is empty to read our first codon in the messenger RNA, GGA. And so out there in the cytoplasm is a transfer RNA with its anticodon of CCU. And so that transfer RNA will bind to it. Once it does that, enzymes will connect our two amino acids together. So we're starting to build our polypeptide or our amino acid chain. Once those two things are connected, still other enzymes are going to come in and remove that first transfer RNA. And so now we've got our two amino acids connected together. We've got our one transfer RNA still attached to its codon. The ribosome then will actually physically move and ratchet or slide down the messenger RNA, moving that transfer RNA into the first position here. Once it does that, now that empty spot is there again first, so our next codon is ready to be read. UGU is our next codon. So the matching transfer RNA with the anticodon of ACA will come in and bind to it, carrying along its amino acid. Once it does, its amino acid is going to be connected to the growing chain. Our old transfer RNA is cut out, and we're ready to repeat the process again. So the process is just going to continue. Our ribosome then is going to ratchet or slide down. Now we're ready to read our next genetic word of AAG. So the transfer RNA with its matching anticodon, which would be UUC, is going to come in and bind to it. Again, when it does, it's carrying its amino acid along with it. Enzymes then are going to connect that lysine to the growing chain, cut out the old transfer RNA, slide down, and read the next word. And again, this is just going to be repeated over and over again as this ribosome slides down the messenger RNA, adding amino acids one at a time. And notice our polypeptide or our amino acid chain is growing, and it has a very specific sequence of amino acids based on our genetic code there in the messenger RNA. So as that amino acid chain grows, eventually we're going to reach a stop codon in our messenger RNA, and that's when we're going to have our last stage of translation, termination. So when a stop codon pops up in that first base of that ribosome, that's going to put an end to our amino acid chain. So the polypeptide or amino acid chain is big enough. It's going to stop this whole process, and that's going to cause that ribosome cap to release in basically the opposite of initiation. All of our chemicals are going to be released and come apart. So here we can see that stop codon popping up in our first space of our ribosome. There is no matching transfer RNA. Instead, there is what's called a releasing factor that will physically bind to that stop codon that will cause termination to occur. And that releasing factor is going to cause all those chemicals to come apart, and that's termination. And now my chain of amino acids is going to be processed. It may be folded up into a specific shape, but we've built our very specific protein. And I think for some of these complex issues like transcription and translation, it really does help to see the moving pieces. And so in this animation, we're going to see initiation, elongation, and termination. Now it's upside down or flipped over compared to the slides we were just looking at. But in initiation, our small ribosomal unit's going to bind to the start codon. Our matching transfer RNA will bind to it, and the large cap will bind up over the top. Once that happens, we're going to go through the long process of elongation. Each codon is going to be read one at a time as they pop up into that ribosome. The matching anticodon will bind holding its tRNA to that. That's going to bring in its amino acid. That will be added to the chain, spit out the transfer RNA. It's going to slide down and read the next codon. And we're going to add the next amino acid, again, one at a time. Each amino acid very specific based on our genetic message in our messenger RNA. That's determining which amino acid is going to come next. And then eventually, we're going to hit that stop codon. When that happens, that's going to cause termination. All of our chemicals are going to be released. Typically, when a cell needs a protein, it doesn't just need one molecule of that protein, but it needs several copies of that particular protein. And as you might guess, this whole protein building process is very expensive in terms of energy and nutrients for a cell. And so what a cell will typically do is make a messenger RNA copy of a gene through transcription, process that messenger RNA, and then that single piece of messenger RNA will make many copies of proteins using many ribosomes during translation. And so what will happen is a ribosome will bind on during initiation and start elongation, start to slide down the messenger RNA building the protein. And then once it's started, a new ribosome will bind behind it at the start codon and kind of follow it down, building a second copy of the protein. And then a third might bind, and a fourth, and so on. And so what we end up with is for each piece of messenger RNA, there may be several ribosomes traveling along it at different places. Over at the right is a ribosome that's almost finished, so it has a long sequence or chain of amino acids hanging off of it. On the left is an ribosome that just started, so its amino acid chain is a lot smaller. And in fact, we have a picture here. This is what this looks like actually under the electron microscope. And it's kind of amazing what we can see today with some of these microscopes, but we can see the chain of messenger RNA actually kind of right down the middle of that slide, and then each of the balls represents a ribosome, and the chain of amino acids you can see coming off of each of those ribosomes. And again, notice the chains are much larger on the right than they are on the left because those ribosomes are almost finished with translation. The ones on the left are just getting started. So let's take a look at an example problem using our DNA and our RNA, and you will certainly see these kinds of problems on the final exam. Now, I will provide a genetic code. You don't need to memorize that, but I do expect you to know how to use it. So, for example, let's say we had this particular piece of DNA. TAC, AAG, GGG, CAT, GAG, TTC. Okay. Can you tell me what is going to be the sequence of the other piece of DNA, that complementary DNA strand? So take a moment and just write that down. And hopefully this is what you got. ATG, TTC, CCC, GTA, CTC, and AAG. Now, if this were part of a gene, and in particular part of an exon that's going to give us protein building information, when this piece of DNA is expressed or turned on, what would the messenger RNA sequence be that would match up with this DNA? So go ahead and pause the presentation and write down our messenger RNA sequence. The important thing to remember when we're doing messenger RNA are two things. Number one, the messenger RNA is only going to have U's in it, not T's, so it's going to be made up of AU, G's, and C's. And second, it has to physically match or pair up with the template. So we're going to ignore the complementary strand, and it matches the template. So to match up TAC, our first three letters in our template, would be AUG. And then UUC, CCC, GUA, CUC, and AAG. Now during translation, then we're going to convert that messenger RNA into protein. And again, this is where our genetic code comes into play. So take a moment here and convert our messenger RNA into the sequence of amino acids that would be in our protein. And this particular chunk would code for the start codon for AUG, then UUC codes for phenylalanine, CCC for proline, GUA for valine, CUC for leucine, and then AAG for lysine. And so again, our specific sequence of DNA has coded for a very specific sequence of amino acids in our protein. So to summarize or wrap things up, DNA structure is made up of 4 nucleotides, our A, T, Gs, and Cs, and we have our complementary base pairing, A's with T's, G's with C's. And they form 2 strands of nucleotides that form that double-stranded helix. And when DNA copies itself, it undergoes that semi-conservative replication, very, very accurate copying. In terms of our gene expression then, our DNA expression, that involves our RNA, which can be messenger, transfer, or ribosomal RNA. Transcription is our copying into our messenger RNA. That messenger RNA gets processed. Remember, we're going to cut out those introns and splice our exons back together. And then we're going to undergo translation, our RNA to protein building stage. And to do that requires initiation, getting all the chemicals together, elongation, and then finally termination. And the genetic code is needed for translation to occur because, again, cells aren't reading every single letter in the genetic code. They're reading the genetic words or the codons in the messenger RNA.